Genes of you and me, genes of you, and we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA. We're all made of the same.
Hello, and welcome to the 20th episode of Discovering New Advances, or DNA, a podcast that keeps you up to date about the scientific world in an easy-to-understand language. So whether you're a student in middle school or a grandparent, you'll easily be able to follow along. I'm Kira, and on this episode, I will give an introduction lesson about gene mapping. For more information, please visit dnapodcast.com or email in at info@dnapodcast.com. This lesson, we're learning about gene mapping. Gene mapping is locating a specific gene on a chromosome to pinpoint its location. It is finding the distance between this selected gene relative to other genes. There are two types of gene maps: genetic linkage maps and physical maps. Let's break down each and identify the differences, starting with genetic linkage maps. The genetic linkage map uses the frequency of genes being inherited as an indicator of the arrangement on chromosomes. This method of figuring out where genes are located based on their inheritance together works because if genes are close to each other physically on a chromosome, they're more likely to be inherited together. During meiosis, chromosomes trade pieces of themselves. This process of switching pieces of the chromosome is called recombination, or crossing over, as the chromosomes cross over each other and swap portions of DNA. Recombination occurs during meiosis when gametes are formed. For those that don't recall, gametes are sex cells such as eggs and sperm. If two genes are on opposite sides of the chromosome, it's more likely they will be separated than if two genes were on the same arm of the chromosome. If genes were on the same arm of the chromosome, they're less likely to be separated during meiosis. These genes would be described as genetically linked. If genes are inherited in a group, they are known to be close together, such as genes together on the arm of the chromosome. I'll use this example again and again. If genes are found on different chromosomes, then their inheritance is independent from each other. Therefore, the inheritance of one gene does not affect the inheritance of the other gene on a different chromosome. So again, let's go back to the idea of genes being found on the same chromosome. If genes are close together, say again on the same arm of the chromosome, their recombinant frequency would be low. Recombinant frequency is how often those genes are separated on different chromosomes at the end of meiosis. If the genes are physically close to each other, this chance is very low. This genetic linkage map uses map units as its key to show the relative map distance between genes. When drawing or reading a genetic linkage map, it is important to know a recombinant frequency of 1% is equivalent to one map unit. Map units are also known as MUs for short. Therefore, 25% is 25 MUs. The percentage cannot go above 50%. At 50%, the genes are located at opposite ends of the chromosome. If you are drawing a genetic linkage map and are given the recombinant frequencies, start with the highest recombinant frequency and label those genes. Knowing this, use that label to write other genes based on where you place the first two. Now, genetic markers can be used like landmarks to figure out other gene locations. These genetic markers are genes or DNA sequence with a known location before the process starts. The other genes on the genetic linkage map are estimated based on the relation with genetic markers and inheritance patterns with other genes. All this information is compiled together to figure out the location of many genes. It is to be noted that genetic linkage was first discovered by the British geneticists Ethel Rebecca Saunders and Thomas Hunt Morgan. It was Morgan who expanded the understanding of genetic linkage by noticing that the frequency of crossing over chromosomes during meiosis differs, and this was the reason genes would be different distances from each other. Now, switching over to physical maps, which is the second type of gene maps. Physical maps take advantage of having entire genomes sequenced. Remember that the chromosomes are just broken up bundles of pieces of DNA. DNA most of the time is in one long strand. This is during interphase, which is the phase the cell is in around 90% of its life. Fortunately, we have sequenced many organisms' DNA, including E. coli, the fruit fly, and even humans. With this information, we can order the genes and figure out the spacing between the genes. Again, these diagrams are known as physical maps. Where do these two maps relate? Glad you asked. The maps match up with each other. Both maps show the same gene-physical relationships. It is not exact by any means, but one map unit on a chromosome encompasses 1 megabase, which is 10 to the 6th power of base pairs of DNA, which is a lot. With this approximation, the two maps can be lined up with each other. These are two different ways to make these maps have the same results. So, two different methods for the same result. Let's review what we've learned. Gene mapping is a way to locate genes on chromosomes. There are two main methods of achieving this. The first is genetic linkage maps, where the relative frequency of the genes being separated are calculated. From this calculation, the genes can be labeled on chromosomes. The closer the genes are on the chromosome, the less likely they are to be separated during meiosis, where chromosomes recombinate by crossing over and swapping over DNA. If they're closer together, they're not going to be separated as easily. Physical maps, on the other hand, take sequenced genomes and label the genes from a pre-existing sequenced genome. The coolest part of this is where the genetic linkage maps, and the physical maps come together. Recall that the recombinant frequency is how often a pair of genes are separated onto different chromosomes. The recombinant frequency equals the map units on a genetic linkage map. One map unit on a genetic linkage map is equal to 1 megabase, which is again 10 to the power of 6 base pairs of DNA. It amazes me that the frequency of genes being separated on a chromosome equals the amount of base pairs there are between the genes on the chromosome. This is a prime example of how math truly is at the heart of biology. I hope this episode helps you understand the concepts of gene mapping. Check out more episodes of DNA Podcast. You can go to dnapodcast.com. There are transcripts on there as well as the audio format, which you're listening to now. There's also a contact form on there for easy access to send in feedback about the show or any questions you might have. But you can also email me directly at info@dnapodcast.com. To keep updated, you can check us out on social media. We're on Facebook and Twitter, both at /DNA Podcasts. Be sure to subscribe to us on iTunes if you haven't already. You can search DNA Podcast and we pop right up. And if you have iTunes open, you might as well just give us a review and rate it. Best way to help us out. So all the links I mentioned, you can go to one link and you can find them all, dnapodcast.com. Thanks for listening and join me next episode to learn and discover new advances in the world of genetics.